Swayam Prabha. Digital India. Educated India.
Good day everyone. The very important aspect of immune system, certainly if I take a straw poll amongst people who are listening and ask you, could you name one disease which is associated with the immune system, chances are that most of them will answer HIV and AIDS. Not surprising, because this is an aspect which has fascinated us since 3 decades or more, since HIV was first noticed and was established in literature. Now, immunodeficiency, which is acquired in nature, certainly in the context of HIV, is a very interesting aspect which we'll try to break down and understand what happens and what is the myth about the disease. Now, HIV, as we know, is an immunodeficiency virus, human immunodeficiency virus. You have HIV 1 and 2. Okay, you could have seen the standard testing basically for HIV, especially if the patient is preoperative screening. You will see them testing for HIV 1 and 2. Now, these are two forms of the virus, and the discussion is common for both forms of the virus. I'll discuss a little bit about the epidemiology of that in the ensuing slide, but remember that they are similar by most accounts. Now, why do you have to learn more about this disease? It is understood that AIDS is the second leading cause of death in men aged between 25 to 44 years, and third in case of women. Now, in an era of increasing prevalence of infections which are life-threatening, tumors, this becoming the second and third leading cause in men and women respectively is a matter of concern, also a point of focus for us to understand. It's a global problem, 40 million living with HIV, 20% itself is in Asia. If you extrapolate this data to an Indian subcontinent, there's a vast pool of people who are having clinical and subclinical HIV. What are the other facets of this? The other side of the coin, treatment options. There are new drugs, ART, HAART are available, but they are expensive. And the cumulative toxicity over a period of time is also a concern. How do you address them? Those are again hot button issues. But where we are concerned, we'll try to understand today the structure of the virus, the pathogenesis of the virus, and let's try to understand some of the infections and tumors commonly associated with HIV. Now, the cornerstone of this is to understand how does the virus infect the cell. We often read or even heard from literature and media that it's almost like a parasite. It takes the host cells and almost incarcerates it. How does it do that? Now, the structure itself, like I told you, there are two forms of the virus, HIV-1, which is prevalent in USA and the European continent, whereas HIV-2 is in the West Africa and also in India, but you tend to see HIV-1 as well, so you test for both basically. We know that it is a retrovirus. By retro, we mean backward. In this context, it means that it can synthesize DNA out of its RNA material. They belong to a larger family called Lentiviridae, and its structure looks something like this. It looks quite complicated, but let's try to break it down brick by brick. Now, you can see that it's almost like a globe. Let's take the context of the Earth. At the very core of it, you will see a small genomic area, which is the core of it. Now, that core is encased by progressive layers, which are called as mantle, which is usually got to do with a lot of lipid bilayers, and then it has an external covering or an external coat, as you can see there, which is in pink, which is usually the membranes which are derived from the host. Now, what are the structures which make up this entire genome? You can see at the core of it, you have what is called as P24 capsid, which is the most prevalent one, and certainly the point of interest for us, because this is the one which they use to screen for HIV, which is at the core of it. You have the RNA, which is an important molecule which is capable of synthesizing a complementary DNA or proviral DNA, as it is sometimes referred to. You have 3 enzymes which are marked here in green, as you can see on your screen. They are integrase, protease and reverse transcriptase, each of them endowed with special properties in the pathogenesis of the virus. Then you have your glycoproteins on the surface, which are very sticky molecules which enable the virus to latch on to the host cells. And then you have some other nucleocapsids such as P17 matrix, which is almost coating the inner core. This also has a role in the pathogenesis. We'll discuss each of this in a lot of detail in the ensuing slides. Now, at the core of it, the proviral genome, as it is called, of the HIV looks something like this. It's a very complicated figure, but try to look at the major points there. The ones which are marked in blue there are three important major genes, which are GAG gene, Pol gene, and ENV gene. Now, the GAG gene is very important, which is the first one on. You can see the first one there on the left of your screen is the GAG gene. It's a major gene because it is important for nucleocapsid binding. So, it's an RNA binding protein, and it is actually a larger molecule which is then processed and made into a smaller molecules, and it has a lot of interactive properties. Pol is a short form for polymerase. It encodes a variety of viral enzymes basically, which could include even reverse transcriptase. That's very vital for the pathogenesis of the disease. You have ENV, it is from the and almost like an envelope gene. This is one which transcribes for glycoproteins. GP120 takes effect from there. GP41, which is also very important, as we'll study later, also are transcribed from the expression of this particular gene. Then you have all supporting players which are marked in pink, like VIF, VPR, VPU, and NEF. Now, these supporting members have a host of responses ranging anything which is very trivial, such as having effect on unidentified host factors, or it can also promote degradation of CD4 cells as they latch onto them. Some of them have roles in the downregulation of surface molecules on the host cells, or some of them even can produce cell cycle arrest at a certain checkpoint, enabling the virus to multiply. So, you can have a range of functions of these accessory genes. The other genes such as STAT and REV, these also are supporting players. Overall, they engage the entire genome of the virus, resulting in a more effective transmission of the disease. Now, pathogenesis, if we want to study, we'll try to understand the targets of these. We know that immune system, that's a given, it's de facto. So, you have the immune system which is attacked by the virus, and of course, in the later stages of disease such as AIDS. Lesser known fact is CNS. CNS also is hit by the disease in some process or the other. The immune system, it usually affects the cell mediated immunity. This we know from our understanding from physiology days, that your cell mediated immunity is the one which is a target for HIV, resultant decrease in the number of CD4 cells and reduced function of the regulatory and the helper cells. So, what happens to the immune system? In the pathogenesis, try to understand that there is infection of these cells itself, which means the virus itself is incorporated into the T cells, resulting in the depreciation or depletion of the cells. Another mechanism is the non-T cells. You would argue that T cells is predominantly present, but it is also true that some of the other players like macrophages, dendritic cells, and Bcells are hampered by the HIV process. Now, we will try to understand them in a little detail. Now, principally with the T cells and its depletion, this is how it convenes. You have the entry of the virus into the cell. Next, you know that it has to engage the host genome one way or the other. That is called as integration. The next, it has to make multiple copies after it integrates, which is called as replication. And finally, they have to break open and release themselves in the bloodstream, ready to attack the other host cells, which is called the release of the virus. Now, these four simple steps result in the depletion of the T cells. How do you ask? You can look up on the schematic representation on your screen there. On the left, you can see the virus structure which we have studied earlier. Now, you can see at the bottom of the virus, you have the GP120. You have the glycoprotein molecules 120 and 41, which have a tropism towards the antigens which are expressed on the CD4 cells. You can see at the bottom of the screen, you have CD4 receptors. As it latches onto them, there is some amount of conformational change within the glycoprotein 120 molecule. This conformational change enables the release or exposure of some of the sites on GP41, which engages the co-receptors, chemokine co-receptors CXCR4 and CCR5. Now, once they engage them, there are more conformational changes, and there is a domain which helps in fusion of the virus to the host cell membrane, which is potentiated. Now, as you can see, as it goes along, there is attachment of the virus to the host cell membrane and resulting in G41 causing permeation. That is, the host cell genome becomes aligned with that of the there is permeation, which means that the genome of the virus becomes integrated with that of the host cell membrane. Now, it's almost like a portal is entering, and there is release of the virus products, principally the genome of the virus into the cytoplasm of the host. So, this is the first mechanism, which is the entry of the virus. Now, once it enters into the cytosol, you can see that the HIV RNA genome becomes very active and very functional, and there is activation of the reverse transcriptase. Now, what does it do? The RNA, which is present as the core of the virus, now begins to transcribe the DNA molecule, which is called as we said earlier, complementary DNA, which is double-stranded in nature, or proviral DNA. Now, sometimes in latent infections, it stops at this level. For a number of years, it may remain very dormant. But very actively multiplying cells, especially given the counts are depreciating, you will see, as you see there on your screen, that the DNA becomes integrated with that of the host genome, which is again facilitated by the enzyme integrase. Now, once there is integration of that, there is transcription. You will see that the products will be released. Usually, they are actually HIV RNA transcripts. So, from the RNA, there was DNA. The DNA incorporates itself the host DNA, and from that HIV RNA transcripts are finally released. As you can see, as they are released out of the nucleus, there is some proteases which breaks down the unwanted molecules, but the very potential molecules, the HIV virions, now have a very core structure. Now, this is what is called as replication of the virus within the host cells. Now, finally, this virus, once it is replicated, it has to release somewhere. Now, they engage the cell membrane of the host, and they take the host cell membrane as an envelope. As you can see, the release of the virus thus happens, engaging itself with the bilayer of the host cell membrane and releases itself into the bloodstream. Now, the bottom structure is your good old virus, endowed with all properties of the outer layer, the mantle, and the inner core. So, this is how the T cells almost acts like a conduit for the release and multiplication of the virus. So, this is what we discussed in the last 5 minutes or so. It's worth reading this part because it's very crucial for the understanding of the effects of the virus. Now, what happens? What is the mechanism of T cell depletion in HIV? We know that it has entered, it has multiplied, and is released. This is what is called as a cytopathic effect. So, direct attack over the host cells. So, what does it do? It has caused increase in the membrane permeability. So, cell loses a lot of its substances, can die, because by osmosis all the contents may be released, and you can have activation of pro-apoptotic genes. So, there is inhibition of protein synthesis, there is upregulation of apoptosis. So, it is a direct cytopathic effect. Sometimes the bystander cells or uninfected cells undergo what is called as activation induced cell death. It is kind of an apoptosis or a programmed cell death, as you have studied earlier. You can have another important mechanism called as pyroptosis. You can go back and read about this. This has to do a lot with the inflammatory release, eventually resulting in the death of the cells. In the reticuloendothelial system, HIV itself can alter the microenvironment of the lymphoid structure. Let us say the tissue architecture in the lymphoid tissues may be altered so as to decrease the sustenance of the T cells in the paracortex. You can have immediate effect over the precursor cells, such as CD4 precursor molecules, resulting in the net loss of these. So, you do not have further differentiation into mature T cells. Syncytia formation. Syncytium is blending of one cell into another, almost like forming a giant cell. Now, this is functionally inactive and results or renders it ineffective. Or instead of just reducing the number, it mitigates some qualitative defect within the function of the T cells, so not effectively synthesizing, let us say, cytokines. It is a qualitative defect. This itself results in the depletion of T cell number, maybe by apoptosis or some other mechanism. Where the non-T cells are concerned, HIV predominantly can infect the macrophages, and macrophages are the cells of interest because the terminally dividing macrophages can get infected and the viruses can multiply within the same. The macrophages almost act like havens because they are resistant to the cytopathic effects of the virus, thus acting like a reservoir and shields the virus. So, this is very important when you institute drugs. Why isn't the viral load decreasing? Maybe macrophages are at play here, shielding the virus. The macrophages and the dendritic cells, which are antigen presenting cells, if you remember, are portals of entry. They act as conduits or portals from where the virus can enter into the body. They also act as transporters of the virus from the, let us say, the skin. into the reticular endothelial system, as we studied earlier. And they can also bring about functional defects such as reduced chemotaxis, microbicidal action being reduced, decreased cytokines, poor antigen presentation. All of these cumulatively cause functional defects in the macrophages, resulting in decrease in number as we go along the nature of the disease. Whereas dendritic cells, again, apart from acting like transporters, they help to maintain the viruses in almost like an intact form. So the virus is prevented from damage from the immune cells by lectin-like receptors. Follicular dendritic cells within the germinal center of the lymphoid tissue almost again act as reservoirs. It is also postulated that the B cells take a hit as well, because there is a lot of lack of function in the B cells. There is evidence that there is polyclonal activation, especially with patients with HIV. If you do a bone marrow analysis, you will see an increase in the number of lymphoid tissue, increase in the number of plasma cells, and hypergammaglobulinemia. Now this is mainly because of an activation of the B cells, and then eventually you can have capsulated bacteria organisms which multiply, resulting in lack of function of the B cells itself. So there is polyclonal activation and lack of function happening simultaneously. CNS, which we alluded to earlier, targets not the neurons but mainly the microglia and the macrophages which cross the blood-brain barrier. You will see a lot of neurological deficits in these individuals, which are again mediated by the products of the virus and the microglia such as interleukin 1-6 and tumor necrosis factor. Glycoprotein 41, which is an essential component of the virus, can induce nitric oxide synthesis within the neurons. And soluble, it is also postulated that soluble JP120 can mediate direct damage over the neurons as well. So if you want to study the latter aspect of this disease in natural history, we know that initially the virus comes in a very innocuous form, which is an acute retroviral illness, where the patient has viremia. So the viral load is very high and the patient comes with very non-specific symptoms which looks like flu. Often they are mistaken for some form of flu, and they will take some form of medication which brings down the infection for the time being. But this may last for 3 weeks intermittently, and they may have let's say a symptom-free period lasting for even months. So this is a typical form of acute retroviral syndrome. As the disease goes along, they enter into a middle phase which is more chronic in nature. This may last up to a decade at best, sometimes let's say fewer years than more than a decade, where there is continuous replication of these virus within the lymphoid tissue. So a very sinister phase where a lot of viral load is increasing, and they present usually with lymphadenopathy. Persistent generalized lymphadenopathy is a typical feature of this phase. And then the full-blown attack over the immune system, resulting in progression to AIDS, where there is complete tattering of the host defense mechanism, resulting in a lot of opportunistic infection and even tumors, what we club together as AIDS indicator diseases or opportunistic diseases associated with AIDS. We will study each of these in a little detail. Just to underline what we have said earlier, the left chart here depicts along the y-axis you can see the CD4 count, and along the x-axis you can see the time frame, that is the years in progression. You can see the first on your left there, it's usually the CD4 count is very high and the earlier phases of the disease numbering let's say 1000 or more. This is an earliest retroviral, that acute phase where you will not see depreciation in number, but as you can see, as it is going along number of years, maybe lasting a decade, there is a period of clinical latency where the constitutional symptoms are not yet there, but you will see that the virus load is replicating very high. As you can see, as the age goes along a decade or more, you will see a lot of opportunistic infections and eventually a death, maybe more than a decade or so. The image on your right shows there is almost a titration between the plasma viremia of the virus and the time frame. You can see that the first indicator that you can have is essentially the viral particles in the plasma, but you need to have a very high index of suspicion for the same. Your next best bet may be detecting the anti-P24 antibody, which is still a basis for testing and screening for HIV by ELISA. Now this P24 antibody will increase from let's say 6 weeks, 1.5 months of the initial infection, and last very long time, a decade and longer, and maybe at the end of second decade it wanes off. So very important test for testing for HIV. The others include anti-envelope antibodies, which begins to rise very early in the course of the disease, and then it plateaus at a certain point, maybe let's say in a year or so, and goes on for a very long time there, but testing becomes problematic. Others include, you can have specific cytotoxic T cell lymphocyte specific HIV peptides, which can do again very expensive and mainly a research domain. Now, this is the currently followed CDC classification, which is very important for us to know in which phase of the disease it is in. This takes into account the absolute count of CD4 cells. As it is seen there, you have 3 categories based upon the CD4 count, which is an absolute count, not a relative count, mind you. If it's more than 500 per microliter, between 200 to 499, and less than 200. Accordingly, you will have 3 clinical categories. You will have what is called as an asymptomatic phase. Initially, again you can club it into A1, A2, A3. You will have C, which is an AIDS indicated diseases, again a very sinister phase. And then there is B, which is between A and C. It's sandwiched between A and C. It's called as symptomatic, which is again divided into B1, B2, B3. Now, it entirely is dependent upon your absolute CD4 count. Now, the last of the diseases is associated with advanced stage of HIV is AIDS defining opportunistic infections and neoplasms. As you can see, they can range anywhere between bacteria, fungus, infestation by parasites, it can be tumors, any load of this. We'll start with the infections. You will see with protozoal and helminthic infections, typically with cryptosporidiosis, they get enteritis, diarrhea, which is refractory, and the load will be very high. If you take a biopsy of the lining epithelium of the duodenum, you will see cryptosporidium within the absorptive villi. You can also have pneumocystis carinipneumonia or girovecii is the subspecies which causes it, very life-threatening pneumonia in the patient, and toxoplasmosis typically affecting the lungs in pneumonia, or CNS, it causes seizures and calcification. You have what is called as fungal infections, very common, especially candida in the esophagus. It causes an ulcer. Do a biopsy, pick up a candida. You can have also it affecting the pulmonary system, or you can have the dimorphic fungi such as coccidiomycosis and histoplasmosis, and you can do a lymph node. You'll see it within the macrophages, teeming inside the macrophages. The bacterial infection range, the poster child for this is tuberculosis in India, and mind you, this is not the garden variety tuberculosis. You can see the avium intercellulare can also do that, or you can have disseminated form of the disease. You can have bacterial infections. Remember, the poster child for this is tuberculosis in India, and this is not any tuberculosis. This is a special one such as avium intercellulare or disseminated forms or miliary forms of the disease, or you can have extrapulmonary manifestations are very common with this sort of AIDS defining opportunistic infection. You can have salmonella related enteritis or even osteomyelitis. Viral infections range from cytomegalovirus, which is very important. You can see the inclusions within the lining endothelial cells. It can cause retinitis and damage irreparably to the eye. It can cause CNS infections. You can have herpes zoster, herpes simplex virus infections, which are localized or even a disseminated form of the disease. And then tumors such as, let's say, lymphomas. In the era of lymphomas, there are HIV associated lymphomas, a separate category on its own. It can cause primary CNS lymphomas, and sometimes even invasive tumors of the cervix in the uterus. So, some of the tumors, just to highlight a couple of slides on it, very common that now that we are in the area of tumors, more often than not you will see that some of the tumors have an intimate association with HIV, 25 to 40% of the cases, because these are usually DNA oncogenic viruses which are very common with HIV, such as, let's say, Kaposi sarcoma, HHV8, human herpes virus 8. There's been a lot of proliferation of your spindle cells, your mesenchymal component, which form the blood vessels and result in this tumor. Lymphomas can range from very systemic non-Hodgkin lymphomas to even primary CNS lymphomas, or even body cavity associated lymphomas, which is a separate entity now in the current WHO classification. This is schematic outline for pathogenesis of Kaposi sarcoma, how the infected cells release cytokines and they attack your mesenchymal spindle cells, and when there is a double hit with your Kaposi sarcoma virus, there will be a lot of growth factors which are released, resulting in endothelial proliferation and angiogenesis. Also, these cytokines engage other inflammatory cells. The net effect is a lot of growth factors which are very pro release of a lot of these endothelial factors, resulting in the tumor which we call as sarcoma. As regards the Bcell lymphomas associated with HIV, you will see the HIV infection which has an effect over your germinal center cells, resulting in hyperplasia of the same, and also a early infection increase affecting your T helper cells, as we have seen earlier. Now, this effectively results in a lot of tumors, mainly because of the mutations occurring during class switch or even somatic hypermutations, resulting in lymphomas of B cells with translocations such as Burkitt, association with CMYC mutation, or large B cell lymphoma such as a diffuse large B cell lymphoma. Some of the virus associated B cell lymphomas are such as EBV associated with Hodgkin lymphoma, such as lymphocyte poor or lymphocyte depleted. You can have primary effusion lymphoma, as we said earlier, and also EBV associated large cell lymphoma. So, these are some of the special categories associated with AIDS. So, that brings us to the end of the chapter on HIV and AIDS. It's important to know the pathogenesis of the disease, the structure of the virus itself, which is integral for the disease, as you've understood in a half an hour or so, and then of course the AIDS defining illnesses, which are very commonplace today in practice, and a good knowledge of this will help us in a long way. I hope you like the class. So, thank you for your patient listening.